Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening, I'm Yvonne Stapp for Science for the Public, and I welcome you to tonight's Contemporary Science Issues and Innovations. Our guest tonight is Zachary Adam, an Agouron geobiology postdoctoral fellow in the Knoll group at Harvard University. He's here to discuss the evolution of the eukaryotic cells, the foundation of all forms of complex life on Earth. Another very exciting area of his research may challenge the assumption that the emergence of life depended on a water medium. His findings suggest that a unique water alternative solvent may have supported the emergence of life, and that's a big excitement. Dr. Adam received his Ph.D. in Earth Sciences from Montana State University in 2014. And he represents the best of the trend by combining his expertise in paleontology, astrobiology, and engineering. He's also founder and director of a group called the Blue Marble Space Institute of Science, and he will be telling us about that as well tonight. We're very pleased to welcome Zach Adam. Welcome, Dr. Adam.
Thank you.
And could you start us by giving us a little bit of foundation about the origins of life? Why is it difficult? and then maybe the three categories, okay?
Right. Well, there are two basic approaches to reconstructing the really deep history of life. And when I say the deep history of life, I'm talking about, before there were dinosaurs, before there were fish, before there were clams or mollusks or any of the kind of organisms that we would recognize today in today's oceans and on the land. Before that, there was this great vastness of time for billions and billions of years on Earth's history, where some really fundamental transitions took place in an evolutionary sense, in an environmental sense, and just about every sense you can imagine. The problem going back is that we have fewer and fewer rocks to tell us about these time periods and these evolutionary transitions. So in trying to reconstruct what happened on the basis of the available evidence, we have fewer rocks to work with as we go backward, and we have a kind of limited assortment of tools in a physical and chemical sense, starting at the beginning and trying to work our way forward. And so what we tried to do is to take these disparate data sets and try to find where they meet in the middle, working from the very deep history forward or moving from the current time backwards.
So we're talking about the emergence of cells, and
Right.
Could you just refresh our memories on the three categories, because I think you work on the complex one.
Sure, yeah, and so that's one of the most. So I studied two of the most kind of profound transitions that are thought to have occurred in the history of life. The first being the emergence of life itself and recognizable cells. And these would be something like bacteria and archaea. They're more or less single-celled, mostly clonal, structurally very simple cells. And then much later on, in about Earth's midlife, if you want to think of it that way, all of a sudden we see these very complex cells showing up. And these were much different than the other two categories of life, bacteria and archaea, and these are eukaryotes. And these are organisms that can really dynamically reshape the external structure of their cells. They have their genetic material kind of sequestered in a special part of the cell called the nucleus. And there are a lot of other kind of complex regulatory mechanisms that allow them to change shape, to have very complex cell cycles. And it was the innovation of the earliest eukaryotic cells that eventually led to us and trees and algae and slime molds and all kinds of other fascinating creatures that eventually came to inhabit the entire surface of the Earth. And so when I look back as part of my work to try to reconstruct the emergence of very advanced complex life, we're trying to say, okay, what was it about archaea and bacteria that helped or may have led to the evolution and emergence of eukaryotes? What was it about the environment that might have been unique that would have driven this kind of very interesting evolutionary innovation?
Okay, so your specialty is the eukaryotes then, and that is roughly, you think that they emerged about when?
Right, so we can kind of run through it. The Earth formed around 4.5 billion years ago. It's not exactly clear when the origins of life occurred, but it was thought to have occurred relatively early. And in fact, as far back as we have kind of really comprehensive packages of rocks, we also see indications that life existed at that time. So that goes back to about 3.5, maybe even 4 billion years ago. The emergence of eukaryotes was followed by, occurred about 2 or 1.5 billion years ago. So there was this really long period of time, 2 billion years, like half of Earth's history, where bacteria and archaea ruled the planet. You know, they ruled the show, they ran the show. And so, and once we see eukaryotes emerge in the fossil record, they seem to have emerged fully developed. We have almost no transition states going from these really simple bacteria and archaea to this monster of a eukaryotic fully developed cell. And so part of the puzzle is saying, what were the intermediate steps? What did they look like? And it's made a challenge, all the more made a challenge today, because the more we look around and we can sequence organisms from everywhere around the world in all kinds of environments, and we really don't see much in the way of intermediate stages of complexity between bacteria and eukaryotes.
I see. So you have to do a lot of like real struggling and inferring maybe to try to reconstruct, because a lot of that would be lost with the geological ages, I guess, in any case. Let me ask you now, one of the most interesting things that you have come up with in your own work is a questioning whether their water, which we take to be fundamental to life. You raise the question, you're not so sure about that. Could you explain why do we assume water is essential for the origin of life, and what you came up with, and then we take a look at your fossils.
Sure. Yeah. So this goes back to the origins of life, and water really is thought to be fundamental to all forms of life as we understand it. I can't get through this interview without having a cup of water in front of me, and that's, what is that, an hour or something like that. And there are, granted, there are organisms that can go a lot longer without water, but as far as we can tell, water is the medium. Water is the solvent that all of our metabolic and genetic reactions take place in our bodies or in any form of life. And in fact, it's to such an extent that we guide our search for life in the universe on the basis of, is there liquid water there or not? So in some of the work that I and my colleagues have been doing, both here at the Harvard Origins Initiative and at the Tokyo Institute of Technology Earth Life Science Initiative, Earth Life Science Institute, we have come across a pathway to creating a solvent that is not water, but that has many of the beneficial aspects of water in terms of a chemical sense, but that overcomes some of the problems with water. So what you may not realize is that while water is important for all forms of life, water is kind of a mixed bag when it comes to being alive in this bag of water right now. So every moment of every day, your body is metabolizing, it's taking in energy, because the water is actually degrading nearly every molecular component of your body at the same time. It's providing a home for it. All of these reactions can take place. There's a paradox there. But at the same time, our enzymes are breaking down in water spontaneously. Our DNA is spontaneously breaking down in water. And if we don't have these kind of error correction mechanisms going on all the time, we quickly will degrade into nothing more than just a kind of pile of gelatinous carbon and a bunch of water. So this idea, and this is based on chemistry that has been done and has been shown over the last 10 or 20 years, this idea is that there are compounds that are better than water for forming polymers. And polymers are making up genes, and they're making up basically enzymes and proteins.
Yes.
So it's been taken as a given that we needed water to originate life because water is in all forms of life that we know.
Yeah.
But if we can demonstrate that there are these alternative solvents, and the one that we work with specifically is formamide, if we can demonstrate a way to produce formamide on the very early Earth when it was just formed in amounts that allow for a lot of chemistry to take place, then theoretically we can start talking about chemical systems that preceded life that were not as degraded by the conditions that water presents for complex molecules.
How interesting. You just said it's been around, the idea's been flirting around here for about 10 or 20 years. It's been speculated. I don't know if that's because they think it's possible on other planets to construct life where there isn't water, but there is some solvent. But
well, it was based on the idea because there are a lot of very talented organic chemists, much more talented than I am, who recognized that compounds like formamide, this alternative solvent, just if you removed the environment and you had never seen a life form before, but you wanted to know how can I make a gene, how can I make an enzyme from scratch.
Right.
These solvents have much better properties than water.
Strange.
And so they remarked on it from the point of view of, oh, isn't that interesting that maybe there are other solvents that would do the job better if we were to try to engineer life from scratch. The overwhelming problem was that there were no pathways to kind of geochemically produce this solvent from the constituents that made up the atmosphere or the ocean. And so what our team has been working on, and the case that we're making, is that there are plausible scenarios where geochemical reactions take place and rocks interact with the atmosphere and with the ocean to create pockets of the Earth where you don't have water at all.
Yeah.
You have formamide and other solvents where you can have much more interesting forms of polymer-based chemistry taking place that are much more conducive to giving rise to life. So the interesting question there becomes, okay, if that's what happened, then where did water-based life come from? And so what we are kind of conjecturing at this point is that it's possible that polymer-based chemistry could have taken place prior to the development of life. They could have reached a level of complexity that was kind of very getting close to life, but really wasn't self-sufficient, and that eventually there could have been some inherent bias or feedback or maybe even like a selective pressure whereby some forms of these chemically interacting systems could survive in water and others couldn't, and that became the basis for life to originate and then inhabit the entire surface of the Earth with all of this available water, having overcome the limitations of water degrading these polymers.
I see. Is formamide widespread? You said something about pockets, but it is, it's, you think of water as ubiquitous, you know, but
Right,
is
yeah.
formamide abundant in that way?
And that's part of the challenge, is that no, it's not. It would have been, and it would have been only found in kind of very specific types of environments at the surface of the Earth. So
It is at the surface.
It's at the surface. So this is a kind of geochemically driven process where you put energy into the system. These organic solvents are created. They're collected in kind of sediment traps or in pockets, but we're not talking about giant lakes of formamide or rivers of formamide or oceans of formamide. It's like puddles of them, or maybe in the way that we think about oil reserves being held in pockets underneath the surface of the Earth. Perhaps we had these pockets of really interesting, very, very complex polymer chemistry going on beneath or right at the surface of the Earth that were in contact with bodies of water, and then that enabled the transition to water-based life as we know it.
I see.
Right.
When you go about getting your fossils, is there any way to infer, you know, that history from a fossil?
I would say it's very difficult because we're talking about a period of time, say
years.
3.5 to 4 billion years ago, when we barely have rocks that survive, just tiny bits of minerals that survive from this period of time. So what we look for are chemical indications from those tiny fragments of available rocks to say, was the environment really hot or really cold? Was the surface environment wet or dry? And then we look at the kind of chemical fingerprints of these minerals to say, were there a lot of terrestrial, like land rocks, or were there basically just this really poorly developed kind of sheet of lava? Because minerals can take many forms, so we want to know how far along had the chemical evolution of the planet progressed. And so that's really the within that very general framework of possibility, we're left to infer what are the likely scenarios that would give rise to complex molecules.
Right. But this is a very exciting alternative view, certainly. And I'm sure it's of particular interest to people doing astrobiology because there are planets and moons even in our solar system where it's quite different, but there is a solvent there. And so there's
always a solvent. As far as we can tell, there's always a solvent.
So that it just makes it much more interesting, certainly. We'll hear more about this, I am sure, in the future. Now, in the meantime, you go looking for these extremely ancient fossils, and I take it that a lot happened geologically and atmospherically and so forth over this long period in the Earth as life is getting started, so it might be hard to reconstruct. But when you are looking for signs of earliest eukaryotes, do you do it with your little hammer and tap on the rocks? Or?
So when we go out to look for fossils of actual life, life that we would recognize, so not an intermediate stage between non-life and life, we have a pretty deep record. At least we're pretty sure that we have a pretty verifiable, demonstrable record of actual life. It may have been simple, it may have been bacteria and archaea, but we've got a record of that going back to about 3.5 billion years ago. And then there was this long period, as I said, of about 2 billion years where not much seems to have happened, or at least it's not recorded very well in the rocks that anything happened for about 2 billion years. We see a massive transition in the atmosphere about 2.5 billion years ago, where we see indications both mineralogically and kind of in the record of certain elements in the rocks that oxygen shot up to unheard of levels. I mean, it was unprecedented in the history of the Earth, and the Earth basically had an oxidizing atmosphere from that point forward, and it's never gone back. And so then there's another period of quiet in the fossil record for another, say, 500 million years. And then all of a sudden we start to see fossils that look like they might be eukaryotes. And what's interesting about eukaryotes is, as far as we can tell, they look to be kind of a chimera. They look to be have genetic and metabolic pieces that are borrowed from both bacteria and archaea. So you can imagine them kind of as this, these are both massive groups of organisms, very complex and very diverse. And yet the underlying machinery is as if you were to take a deck of cards and shuffle them together, you'd be mixing and matching different metabolic and genetic components to produce a eukaryotic organism that was capable of so much more than the mere sum of its parts. And when we go out into the field, we're basically looking for indications when we're at the outcrop that life has somehow manipulated or influenced the sedimentation of the rocks that we see.
That is most interesting. And so when you go out to the rocks, where do you go? What's a good place to go?
Well, I'm biased because I did my work in west and central Montana, which has a very distinguished history in terms of its paleobiology. So there was a man named Charles Walcott. He was the man who discovered the Burgess Shale, which has given us so many insights into the emergence of animals from rocks up in Canada. But he worked on these rocks, and at the time they had very little concept of exactly how old the Earth is. They knew that there were all these rocks that had a very rich fossil record of animals and plants, going back only so far in time, and then before that, they basically just said, time X, time Y, time Z. We don't know how old it is, but we know it's much older than animals because there are no animal traces to be found. And try as we might, we don't see any recognizable organisms. And what happened was that he was studying the rocks of Montana, and he noted about 120 years ago that there were these funny traces on the rocks. And he didn't really know how to make sense of them. He saw them, he had been, this was a time when you're really trying to figure out the basic structure of geology, how rock units relate to one another. And he knew, based on everything that he had seen in all of these rocks of all these different ages, that the traces were almost certainly biological, but he didn't really know what type of life it actually was. So he basically did what we all do when we see something confounding. We write up our description of it, we take our best guess at interpreting what it is, and then we put it out to the community to see what they think. And I've actually brought a sample this is a very famous locality for Precambrian paleobiology. And the fossil type that he saw has subsequently been named Gripenia spiralis. And I've brought some samples here, and I don't know if we can see. Here we go. So I've put a little helpful arrow at the bottom, and basically it's this little round spot on this very flat piece of shale where it looks like there's a coiled ribbon that's been kind of compressed and preserved on the surface of the shale. And what it looks like when they first saw it and tried to make sense of it, it looked like, you know, a child had come and scribbled with a crayon or a pencil and just drawing these little marks on the surface of the rock. And they realized there was a very regular structure. It has a constant width, it has a more or less constant length, and that it was more than just the random scribblings of a child.
Yes.
And what he thought at the time was that this was a trace fossil of a much larger organism that had kind of scraped its way along the surface of the sediments, which flew in the face of all established science at the time. You know, it was a very far out idea to say that over this incredible spans of time, you'd have very complex organisms that are capable of walking across a sediment water interface to leave this kind of footprints, if you will, in the trace of the mud without any trace of their body. So this and a number of other controversies kind of stewed for a very long time in the field until the evidence became overwhelming that indeed there was life at the time, it's just not of the body type that we would associate with animals. And so there is this evolutionary history that vindicates Darwin's views that animals had been preceded by organisms of, you know, gradually increasing complexity over a much deeper history of Earth's history.
Isn't that something? I take it that the area that you investigate in Montana, it's not that kind of fossil record is not abundant across the Earth. I mean, that's selected places. So Montana, and where is it in Montana, the range?
Right. So there are a number of places, and basically all along the border between Montana and Idaho, you get these rocks called the belt supergroup that show up beneath rocks of, you know, that have things like bones and plant fossils and things like that. And they basically show up everywhere from, if any of our viewers have been to Glacier National Park and seen the magnificent cliffs and just these massive exposed layers and layers and layers of sediments that are nearly unbroken, and you can walk right up it like steps for literally miles in height or kilometers in height.
And you're going back in time.
And you're going back, you're 1.5 billion years ago in Earth's history. And those rocks really crop out throughout all of the area, going from up near the Canadian border almost down to Yellowstone National Park. So it's really almost the entire western edge of the state.
So for whatever is exposed, that's very rich for this kind of a history, because it's not preserved everywhere on Earth, right?
And so there are probably half a dozen really good localities that preserve the traces of these fossils around the world. I got interested in this project because when I looked at a map of where all of these localities were, I saw that they were in very difficult to reach places. I mean, they're in Siberia, they're in central China, they're in northern Australia, they're in northern India. But I, as a graduate student, I can't go to those places to look for life. And I had known that there was this history of the paleobiology of the belt supergroup in Montana because of fossils like those that had been described, like Gripenia, stromatolites, which are these kind of domed accretions of sediments that are indicative of biological activity. And so what I wanted to do was to say, okay, within this very large body of rocks, are there other traces of fossils that might provide more insight into the origins of eukaryotes? And there had been some work by a man named Bob Horodisky. He had worked on trying to dissolve away some of these rocks. And when you dissolve away the rocks, you're left just with the organic detritus, the junk that's left over. And if you dig through this detritus carefully enough with a microscope, sometimes you find microfossils. And so, unfortunately, he had passed away unexpectedly, and no one had really picked up his work. And so I designed a project to kind of pick up where he had left off, to try to look for microfossils in parts of the belt supergroup that had indicators of life, such as Gripenia spiralis or stromatolites. Yeah.
How about that? And you were successful.
Yeah, and it's a relatively easy process. You are in the field and you're looking for the kind of rocks that really fall apart easily. When you're in Glacier National Park, you can climb these magnificent cliff faces because the rock is it's a very hard rock. It's very lithified. And I was looking for the opposite of that kind of rock. I want muds that just fall apart in your hands. And so the outcrops tend to be much less magnificent. They tend to be on the sides of roads or in ditches or wherever water is kind of cut through. And they're about
not
this
the
tall. They're not very
places.
glamorous. If you're a flea, it looks fantastic. But if you're a human, you're stepping over it.
Yeah.
And so you go to these sites. You look for these types of rocks with these properties. You dissolve away the host rock with hydrofluoric acid. Anyone who watches Breaking Bad knows about hydrofluoric acid, and that's very dangerous. And then you dissolve away the host rock, and then you dig through what's left, and you find these magnificent fossils that have, like I said, eukaryotes are renowned for having this very dynamic ability to reshape their cell shape, to take on many different forms, to form walls or doors in the opening of their cell wall, and then to close them up again. And that's exactly what we found when we looked very, very closely.
That's amazing. And so you brought some examples, I think, of from your most recent trip. That's from this summer. And can you show us? Can you tell us about them a bit? Okay.
So, and this kind of gets in on this last trip. We were trying to really get firm dates, like exactly how old are these rocks. And so while we were visiting the outcrops, it's very rarely that you find any specific indicator of life without finding other indicators of life. So actually, if you go back, for example, to the site where Gripenia, the little scribblings on the rock, are found, if you look closely, you'll find other indications that life was there. And so, for example, if you look at this specimen, this is actually called an elephant skin texture. And it basically looks like elephant skin. Like if you
Yes,
were,
it does.
you can see that there are some folds. Let's see, right down here on the bottom.
Yes.
It looks like there was if there was a very thin tablecloth that was sitting on this surface, and you were to push downward over here,
Yeah.
it would kind of bunch up in these little folds down at the bottom. And you can see some others that are on this side as well. So basically, what this looks like when we're looking for indications of life in the field, what this looks to me, it looks like microbes there had been a very fine surface of mud at the bottom of this body of water. Microbes inhabited that interface between the water and the mud bottom and kind of solidified it, made it into kind of a glue. And then maybe some currents came, changed direction to push that around a little bit, just like if you were pushing a tablecloth on a table, or maybe the slope adjusted slightly and so some of it was slumping down, and it created these kind of fold textures in the surface. It's not a direct indication of life, but it's a kind of an indirect or an associated signature for microbial activity.
And a trained person can distinguish between ripples, say, that are in rock and an indicator like this, that life was here.
Right.
Okay.
Right, and you can see the relief on these is
different.
very, it's a very low relief. You have to be kind of looking for it in detail. And these are also asymmetrical. Like ripple marks usually are going to be very symmetrical and very repeating.
Right.
These are asymmetric, and they have kind of these chevron folds where it looks like you've just kind of pushed the tablecloth off the table.
Right.
Yeah.
Is this any particular, an indication of any particular microbe, or it's just undefined?
This one is probably just undefined.
Okay.
It's more or less probably just a bacterial or archaeal culture.
Okay.
But if you were to go back, what this would say to someone in the field is that, okay, if you have all of these other traces of life, like Gripania, like these kind of microbially influenced structures or surfaces, you would stand a reasonable chance, if you wanted to focus your efforts to find microfossils, you would want to look in this particular rock or this particular rock type.
Okay,
Right.
all right.
Yeah.
And so you came up with some other goodies?
So part of the challenge of this, is that you don't always know what you're looking at, and sometimes the jury can be out for a very long time as to whether you're dealing with an actual fossil or a dubio fossil or a pseudo fossil, like something that you're just not sure about. So there's another type of fossil that is found up in Glacier National Park. It's called Horodischia. It's named after Bob Horodisky, and it basically looks like if you had taken kind of a string of beads and kind of gently pushed them into the mud surface and then lifted it up. They're these kind of round shapes separated by equal distances along a line. And I happened to be out in the field, and the sun was at the right angle, and the sediments had kind of eroded away in the right way. And so I actually found another type of fossil that looks like it might actually be a specimen of Horodischia. But you can see all of these. You can see all of these little pits in the surface here, and you can see that they're all more or less kind of equally sized. And the problem is that there are so many of them here. It's really difficult to resolve whether or not each of these tiny little pits are actually in a line, or if it's just a bunch of equally sized compressions in the surface. So this would be something that, I would hesitate to call this an actual fossil. I would have to go back and sample it quite a bit more and gather a lot of statistical information about it to say
I understand,
whether
but that's
it was
part of the
life or not.
process, I guess, isn't it? But it's interesting, to the untrained person, you walk right past it. You wouldn't even notice it.
Yeah.
So if you are trained to look, it's a different process altogether, isn't it?
If that's how you're spending your day out in the middle of nowhere looking at rocks,
you better
that
find
no one
something.
is looking at.
That's right.
So, but another indication here would be that the rocks that are in Glacier National Park are roughly the same type of rock and almost exactly the same time period as this rock. So by inference, you have some basis for saying, okay, I should be here looking at these, and I would expect
Yeah,
to
I
see
see.
something like
I
this.
see.
But it's 300 miles distant from this particular outcrop.
Ah,
Yeah.
but all in all, that area must be very rich in this very early period of fossils,
Right.
if you know what to look for. Is this shale? Or
Yeah,
is
all of
this
this
shale?
is-
Because it just looks like it would come off very easily. Can you just sort of slice off pieces and have a look
Yeah,
underneath all the time?
I'll take a geology hammer in the field, but oftentimes if it's the rock that I'm looking for, I really don't need it. I can either, for these particular specimens, I can just kind of pull them off the outcrop with my hands, because I'm not looking for the really well lithified, tough rock. I want the softer stuff.
Is where you would look in any of these hotspots on Earth for these kinds of fossils?
Yeah, generally speaking, stromatolites tend to show up in relief when the rock is really well lithified. So in Glacier National Park,
Right.
again, it's renowned for its stromatolites, these magnificent columns that
Right.
are many meters tall and many meters in relief, and they show up because they're on these really stark cliff faces where you can see each line on the side.
Isn't that something?
Yeah. So there's depending on exactly how you're looking and what fossil type, the history of the rock may preserve different types of fossil traces than others. So microfossils are great in the soft shales, but fossils such as Gripenia, which rely on you seeing them in relief with these kind of large plate-like exposures, the rock needs to be a little bit more cooked to show up like that at the outcrop and to be visible to someone who's looking at them.
I see. And you're likely to find, I just want to verify that these kinds of fossils might be in similar layers around and these selected places
Right.
on the Earth,
Right.
and kind of an indicator of a particular time period, perhaps,
A
or
particular
whatever.
time period and a particular environment, and perhaps a particular kind of history of preservation of the rock itself.
Okay.
Yeah.
I would like to get one other thing sort of clear.
Sure.
When you're looking at these ancient eukaryotes, are they quite different from what you would see in a modern eukaryotic cell?
So that's part of the challenge of our work. These particular rocks are about 1.5 billion years old, and 1.5 billion years is a lot of time
Yes.
to evolve from one thing into something else. If you give me 1.5 billion years, I'm sure I'll evolve into any number of creatures that you can imagine, just given the time. And it's the case even more so for small organisms, because they have a very fast turnover rate, they have a very fast kind of lifespan.
Yes.
And so the generational time to manifest evolutionary change presumably is a lot faster. Part of the puzzle, though, is that when we look at fossils that are of this age, they don't seem to change for hundreds and hundreds of millions of years. So why didn't they change? Is it just apparent? Like, is the morphologies that are preserved, do they just tend to be of one particular type or a small subset of types? Or was evolution just fundamentally different somehow? Was the rate of evolution different because the environment was different? Or there was some missing genetic component that allowed them to change with greater frequency? These are all open questions. And so when we try to compare the fossils that we see to things that we see today, we have to be a bit skeptical about equating morphological similarity to genetic similarity or species similarity.
I see.
But we might be able to extract very specific kinds of clues. Like if they grew, for example, by shooting and growing tubes out of their surface, that's a very metabolically expensive way to grow. So you must be getting a lot of nutrients back. So you
I
can
see.
say something about, was it perhaps photosynthetic and increasing its surface area?
Yeah.
Or was it trying to absorb nutrients like a fungus would? Like when you see the fuzzy growths over fruit, was it something that was more absorptive in its metabolic strategy? So you can draw these kind of top level inferences, but you're very limited by how much you can make that comparison.
I see, and it makes sense that might be a question for some time, because
Right.
reconstructing at that level is really quite difficult. I would imagine there'd be things like astrobiology, fields like astrobiology, that would be really salivating about what you're doing here. Is that
Right.
true, and are you interested in that also? Maybe you better tell us what that is.
Right, yeah, so astrobiology is the search for life in the universe, and we are at a very special time. Like, even when I was in middle school and high school, at that time there were 9 planets in our solar
Isn't that
system.
something?
Then there were 8 because Pluto is a dwarf planet. But that's it. There are a lot of moons, but those are the only planets that we know about. And now we're literally at the time where announcements of discoveries of planets around other stars, they come in batches of 10, 100, or 1,000, depending on the data set.
Every week, it seems.
It seems like every week or every month there's a new exoplanet discovery, a planet outside of our solar system. So NASA, in all of its foresight, anticipated the discovery of lots of planets outside our solar system, and they want to know, and this gets back to the fundamental transitions that I study, what are the conditions that life is going to originate on any number of those other planets, and if so, what is its probability? So what fraction of those planets have life on them? And B, what fraction of those planets that have life are likely to have complex life, advanced life? And then you can go up to higher levels and say, okay, what's the probability that they'll be technologically complex or something like that? But understanding the constraints or the drivers of the emergence of life or the emergence of eukaryotic life are really fundamental to understanding whether it's half of those planets with life, or 10%, or 1 in a million, 1 in a billion. And so closer to home, we look for these traces of eukaryotic life because, or any life really in the deep fossil record, because we want to know if we're, for example, sending very sophisticated rovers to the surface of Mars, and we see all of these beautifully preserved sedimentary structures. The scientists and engineers in control of that rover want to know, do I go to that outcrop or do I go to that outcrop? And when I go there, which part of the outcrop do I focus the camera on? And if the outcrop is, a sediment that is more or less kind of flat and crumbly versus kind of tilted up and kind of platy and well exposed, what types of biosignatures am I going to look at this outcrop versus the other? And it really gets back to the nature of how these particular fossils and others like them, how do they show up at the outcrop? And what's the probability that if they're right in front of us that we're going to be able to recognize it given the resolution of the camera?
Exactly.
So
there's
even
a
here,
direct link.
it's a very
here.
direct link. And even here, when I'm holding up specimens for the camera to try to resolve what the features are, and we have fantastic lighting, and we have state-of-the-art camera technology, and we have very talented camera crew working everything to get it just right. When you're beaming these images from millions of miles away, it gets really challenging really quickly, and you want to make sure you're looking for the right traces of life.
Yes, that is really neat, and what an exciting time to be applying this. Before we end, we're about out of time. I'd like to ask you about these organizations that you are a part of. If you could just give us an idea of what they do.
Sure. So the Origins of Life research, I'm not doing this alone. I'm working with some really talented people, both here at Harvard in the Origins of Life Initiative, and in Japan at the Earth Life Science Institute. So on the research side of things, I've worked with a number of people in terms of outreach, because as we've seen with the cutting of a lot of budgets in scientific institutions, the funds available to do outreach and to explain, just really at a basic level, what is it that I'm doing when I'm spending weeks out in the woods stumbling around looking for rocks, and explaining to the public, like, why is it important and what's the kind of benefit that they're going to get, those funds have been reduced substantially. And a group of my colleagues and I started an organization called Blue Marble Space, and we basically put our extra time into communicating to the public exactly what we do, how it benefits them, and what some of the either downrange, cultural impacts of this work is going to be. I mean, asking about where life came from and whether there's life in the universe is possibly one of the oldest scientific questions we have. And so it's not a trivial thing to invest time into studying that. Right. So our objectives are really just to explain to people and to take the time to try to increase our ability to explain to people what we're doing and why it's important.
Well, you just did a super job. Thank you so much. And it is good to remind the public too about the terrible cutbacks in a lot of basic research, research in general, and I hope we will see a change in that. But I certainly agree with you that this is having a seismic effect on our global culture as people become aware of this and relate what you're doing to outside the planet, which is a very recent thing. I mean, the space aliens have come and gone many times. We don't hear about it anymore, but now there are these more really serious questions. It's really great what you're doing. Thanks.
And if I may point out, there are unforeseen and unforeseeable applications of this basic research. For example, in trying to reconstruct the origins of life, we're limited by the types of molecules that are found in life to try to see how they change their environment. If we could reconstruct new forms of life, we could possibly be engineering them to solve very basic problems. We could be engineering a form of life that draws CO2 out of the air and does not do any other metabolic activity to help mitigate climate change, for example. That's not something that you think about commonly when you think about origins of life, but there are definite technical possibilities that will better our lives to fall out of fundamental research.
That's really an interesting point. Thanks for bringing that up, because we don't usually think like that, but with all of the biological engineering going on, that's certainly an interesting area. In any case, it's just fascinating in every respect. And Dr. Adam, thank you so much for joining us tonight.
Thank you for having me. Yeah.
Okay.